Yeah. Hello, my name is Rada Koldamova, and I work at the University of Pittsburgh. Our group is focused on Alzheimer's disease and explore the connection between lipid metabolism and Alzheimer's disease. In this presentation, I'm going to talk about the role of ABCA1 transporter and apolipoprotein E in Alzheimer's disease. ATP-binding cassette transporter, A1 or ABCA1, is located in plasma membrane and mediate cholesterol efflux. This is a complicated process. In the first step shown here, ABCA1 export cholesterol and phospholipid to the outer part of the membrane, and in the second part, the second step, these lipids are transferred to lipid-free apolipoprotein A1 or apolipoprotein E, and in the third step, discoidal HDL or high-density lipoproteins are formed. ABCA1 is essential mediator for the formation of high-density lipoproteins, and when functional ABCA1 is missing, the level of HDL is almost zero, and there are additional consequences of ABCA deficiency because non-lipidated apolipoproteins A1 or E are usually unstable and degrade fast. So in cases of ABCA1 deficiency, not only HDL are lacking, but the level of apolipoprotein A1 is virtually zero, and APOE is very low. ABCA1 and also apolipoprotein E are transcriptionally regulated by ligand-activated Liver X Receptors or LXR, or Retinoic X Receptors or RXR. I'm sure most of the people present at this talk know what is Alzheimer's disease, but I will just briefly describe it. It is a senile dementia characterized by cognitive decline, and advanced age is the strongest risk factor for the disease. Morphologically, the brain of Alzheimer's disease patients is characterized by extracellular deposit of amyloid beta that represent amyloid plaques, stained with green on this slide, or intracellular depositions of hyperphosphorylated protein tau, also called tangles. There are two forms of the disease. Actually, there is a late form of the disease called also the sporadic form that arises usually at advanced age. And there are familiar forms of the disease caused by mutation in certain genes, mostly the precursor of amyloid beta, or presenilin 1 and presenilin 2. More than 90% of the cases are late-onset sporadic disease, and there is no obvious cause for it. On this slide is shown the processing of amyloid precursor protein and the generation of A-beta peptide. So amyloid precursor protein is transmembrane protein. It is cleaved by two enzymes, two secretases, beta secretase and gamma secretase, which generate amyloid beta peptide. It is secreted as monomer, but because it is highly hydrophobic, it oligomerizes and eventually forms bigger aggregates that form the plaques. A-beta is usually around 40 to 42 amino acids long, and the longer forms of the peptide form aggregates more readily. On this slide are shown micro photographs of different stages of A-beta aggregation. Here is shown a mixture of A-beta monomers and oligomers. On B are shown protofibrils, and on C bigger aggregates, fibrils that usually comprise the amyloid plaques. Monomers and the monomers and oligomers usually are called soluble form of amyloid beta, and fibrils that form the plaques are also called insoluble amyloid. Recent data suggest that soluble A-beta oligomers are more dangerous for cognitive function. Because A-beta affects cognitive function, it is important to overview the mechanisms of A-beta clearance from the brain. As I mentioned, A-beta is secreted as monomer, it forms oligomers, and finally fibrils. And there are several major pathways for its clearance from the brain. One is efflux, or clearance via brain-blood barrier. It is mediated most probably by LRP1. It is low-density related protein 1, and maybe other receptors shown here could participate. Another way of A-beta clearance is degradation in the brain by extracellular proteases, such as insulin-degrading enzyme or neprilysin secreted by glial cells, or phagocytosis by microglia. And finally, part of A-beta could go to cerebrospinal fluid and be cleared by drainage into perivascular arterial space. Why are we interested in ABCA1? This transporter was identified in 1999 as the gene mutated in Tangier disease. Tangier disease is a rare monogenic disorder caused by loss of function mutation in ABCA1. It is characterized by extremely low HDL and APOA1 level. ABCA1 also mediates cholesterol efflux from glial cells in the brain and is responsible for the generation of brain HDL particles. When ABCA1 is lacking, the level of protein apolipoprotein E and A1 in the brain, as well as the HDL particles in brain and cerebrospinal fluid, is significantly reduced. ABCA1 deficiency was found to increase amyloid deposition in AD model mice. On this slide, I'm showing brains of mice, APP transgenic mice, expressing wild-type ABCA1. Amyloid plaques are shown in green, and these are APP mice with targeted deletion of ABCA1, and you can see that there are a lot more amyloid plaques. It was reported by our and other group, David Holtzman and Cheryl Wellington group, in 2005. So why are we interested in APOE? You probably know that the inheritance of APOE4 allele is the major genetic risk factor for late-onset Alzheimer's disease. There are 3 major isoforms of APOE. Here are shown the major amino acids replacement in different isoforms. APOE3 is the major allele. It is expressed in more than 80% of the population. E4 is expressed approximately in 40% of population, and E2 is really rare. So as I said, the inheritance of APOE4 increases the risk of Alzheimer's disease. In contrast, the inheritance of APOE2 decreases the risk of Alzheimer's disease. So in the next few slides, I'm going to show you the result of an experiment where we ask the question how the deficiency of ABCA1 affects Alzheimer's disease phenotype in APP transgenic mice that also were made to express human APOE3 and E4 isoforms. So these mice express mutated human amyloid precursor protein and presenilin 1. And in this experiment, we compare the phenotype of the mice with 1 or 2 copies of ABCA1. Mice were examined at age of 7 months when the phenotype in this model is fully developed. So first question we asked was how the protein level of APOE and APOA-I in the brain is affected by the deficiency of ABCA1. Here is shown a graph representing APOE level, protein level in the brain in E3 and E4 mice. And you can see that the deficiency of 1 copy of ABCA1 decrease APOE level in E3 and in E4 mice, but the effect is much more pronounced and statistically significant in E4 mice. Similarly, APOA1 level was decreased by the deletion of 1 copy of ABCA1 in E3 and in E4 mice. And again, the difference was more pronounced and statistically significant in E4 expressing mice. So the conclusion from this experiment is that the deficiency of even 1 copy of ABCA1 decreases protein level of APOE and APOA1 in the brain. In the next experiment, we tested how the deficiency of ABCA1 affects cognitive performance of APPE3 and APPE4 mice. We use a radial water maze paradigm shown here, which tests spatial memory acquisition of mice. And on the first graph A is shown a comparison between APPE3 mice having 1 or 2 alleles of ABCA1, and you can easily see that there is no difference in the performance. In contrast, when E4 mice were compared between ABCA wild type and heterozygote, you can see that the performance in radial maze is significantly different. A red line represents ABCA1 heterozygote mice, and they perform much worse than the ABCA1 wild type E4 mice. So the conclusion from this experiment, also others which I do not show, is that the lack of even one copy of ABCA1 significantly impairs, aggravates cognitive deficits in APOE4 expressing mice, but not of E3 mice. In the next experiment, we were interested to assess what is the level of amyloid plaques and how the deficiency of ABCA1 change amyloid deposition in E3 and E4 expressing mice. So first, this figure here compares APPE3 and APPE4 mice expressing wild-type ABCA, and you can see that there is approximately 2 times difference between them, which is reminiscent to human population when E4 carriers have more amyloid plaques than E3. When one copy of ABCA1 is deleted, you can see that the plaques in E4 mice increase more than 9 times, and there is very significant difference between E4 and E3 mice. The other figure on B shows also amyloid plaques. However, here they were stained with X34 stain that visualize the compact amyloid, and here with anti-amyloid antibody that also stain and visualize diffuse plaques. So the measurement of A-beta positive plaques also confirm the previous finding that the deletion of one copy of ABCA1 significantly increases amyloid plaques in E4 mice, but not in E3 mice. You can compare this column with this column, and you'll see that there is practically no significant difference between them. Similarly, if you compare gray to green, you'll see that there is no difference. In contrast, if you compare light green to orange, you'll see that there is significant difference, which tells us that the deletion of one copy of ABCA1 or the deficiency of ABCA1 differentially increases amyloid plaque in E4 mice. To confirm these studies, we extracted insoluble amyloid from the brain of the same mice, and we measure insoluble A-beta 40 and A-beta 42 using ELISA. Essentially, this experiment confirmed the previous with amyloid load, and you can easily see that insoluble A-beta is statistically significantly increased in APPE4 mice heterozygous of ABCA1. In contrast, there was very little difference between E3 mice with wild type or ABC heterozygotes. So the conclusion from this and the previous experiment is that the deficiency of one copy of ABCA1 significantly increases amyloid deposition only in E4 expressing mice. In this experiment, we wanted to test how the deficiency of ABCA1 affects clearance of A-beta. For this purpose, we inserted a cannula in the brain of the mice in hippocampus, and we use microdialysis of freely moving mice to collect interstitial fluid and to measure A-beta level in this fluid. So on A is shown the steady state level of A-beta-42 in interstitial fluid. And here are compare APOE3 mice with wild-type ABCA1 and ABC heterozygotes. You can see, if you compare gray to yellow, that there is very little difference, and it is not significantly different. It is not statistically significant. If you compare E4 mice with wild-type ABCA1 and ABC heterozygotes, you'll see that there is statistically significant difference between the level of A-beta-42. On B is shown half-life of A-beta-42 in interstitial fluid, or how fast A-beta disappears from the brain. Again, on E3 mice, there is slight increase when ABCA1 one copy is deleted, but this difference is statistically not significant. In contrast, in E4 expressing mice, there is statistical significant increase of A-beta-40 in heterozygotes mice when they are compared to wild-type ABCA mice. So the conclusion from this experiment is that the lack of ABCA1 exacerbates the removal of A-beta from the brain, and it is more pronounced and statistically significant in E4 expressing mice. Lastly, we measure the level of APOE and high-density lipoproteins in plasma of E3 and E4 mice. On A is shown APOE level, and on B, HDL level in plasma. And you can see that in E4 expressing mice, there is a very big decrease of APOE and very big decrease in HDL concentration, which is exacerbated by the lack of one copy of ABCA1. So let's say on this graph, if you compare HDL level in E4 mice, ABC heterozygotes, to E3 mice, ABCA1 heterozygotes, there is almost 40% difference, and this difference is statistically significant. Lastly, we check the correlation between X34 plaques and HDL in the brain and HDL level in plasma, and you can see that there is a statistically significant correlation between these two parameters. This correlation was negative, meaning that when HDL level increases, there are less amyloid plaques in the brain. This experiment suggests that there might be a causal relationship between plaques and HDL level. So the conclusion from this study is that ABCA1 expression is essential for APOE protein level in the brain and regulates its lipidation. Lack of one copy of ABCA1 differentially increases A-beta plaques in APOE3 and E4 mice. Actually, it means that it increases A-beta plaques in E4 mice and does not have effect on APOE3 expressing mice. The deficiency of ABCA1 decreases A-beta clearance from the brain, and the effect is more pronounced in E4 mice. The level of APOE and HDL is more affected in E4 mice, and the level of plasma HDL correlates to amyloid plaques in the brain. We conclude from this that APOE4 mice and probably E4 carriers are more vulnerable to the effect of unfunctional ABCA1 and probably to additional genetic defects. The next experiment, in the next few slides, I'm going to talk about the effect of RXR agonist bexaroutine on AD, Alzheimer's disease phenotype in mice expressing human APOE isoforms. Bexaroutine is, as I said, Retinoic X Receptor agonist. It is FDA-approved drug for treatment of cutaneous cancer. Previously, it was shown by Gary Landreth group that bexaroutine upregulates ABCA1 and APOE level in the brain and clears amyloid plaques. They also show that the treatment improves cognitive deficits of mice, APP mice expressing mouse apolipoprotein E. We extended these studies by examining or assessing the effect of this drug in APP mice expressing human APOE isoforms, E3 and E4. We treated the mice at 7 months of age for 2 weeks, given orally, and evaluated their cognitive performance and amyloid load. So on this slide is shown the effect of bexaroutine on cognitive performance in E3 expressing mice. With the black line are shown non-transgenic APP mice, and their cognition is considered normal. With red line is shown APP E3 mice treated with control mice treated with vehicle, and with green line is green line shows the performance of APP E3 mice treated with bexaroutine. So you can see that mice treated with bexaroutine have improved performance, which is statistically non-significant from wild-type non-transgenic control, and is statistically very significantly different from mice treated with vehicle. On B is shown the effect on APP E4 mice, and again with black line are shown non-APP mice that don't have amyloid plaques and are have normal cognitive performance. With red line are shown control APP E4 mice treated with vehicle, and with purple APP E4 mice treated with bexa. So you can see in this experiment that purple and black are almost coincide, which means that bexaroutine restore cognitive performance of APP mice, and they were significantly different from the mice treated with vehicle control mice. We confirm this result using additional tests. One of them is shown here. This is called novel object recognition test, in which mice are trained to recognize novel object, and the next day, the testing day, the percent time spent with the novel object is recorded and is considered as measurement for learning. So on the graph first on this side is shown the performance of APP E3 mice, and they are compared to non-transgenic controls. These non-transgenic controls express APOE E3, but they do not express APP transgene and do not have amyloid plaques. So as you can see, control APP E3 mice are significantly different from their non-transgenic littermates. When they were treated with bexaroutine, the performance significantly improved, and it cannot be distinguished from their non-transgenic littermates. Similar data were received with E4 expressing mice. Here are shown non-transgenic controls, and this is APOE4 mice, control mice treated with vehicle. You can see that there is statistically significant difference between them and the control non-transgenic mice. When they were treated with bexarotene, there was complete restoration of their cognitive function. So the conclusion from this and the previous test is that bexarotene restored cognitive decline in APP mice expressing human APOE3 and human APOE4 gene protein. Previous data from another group demonstrated that bexarotene clear amyloid plaques. So our next goal was to see the effect of amyloid plaques and insoluble amyloid. And on A is shown the result from examining amyloid plaques. We did not find that bexarotene affected amyloid plaques in APOE3 mice, neither APOE4 mice. We also examined insoluble A-beta, which is not shown here, and this confirmed that bexarotene in this particular experiment did not affect amyloid plaques or insoluble amyloid. On B is shown the effect on soluble A-beta oligomers. So soluble A-beta was extracted from the brain, and the level of oligomers tested with antibody L11 from Charles Grape lab that specifically recognize oligomers. We found that treatment decreased soluble oligomers in E3 expressing mice and in APOE4 expressing mice. And the conclusion from this and other studies is that bexarotene did not change amyloid plaque, did not change insoluble amyloid, but decreased soluble A-beta oligomers. Our next experiment was to examine how A-beta in interstitial fluid is affected by bexarotene treatment. So on A is shown microdialysis. It's A-beta collected by microdialysis and ELISA for A-beta 40. You can see that treatment significantly decreased A-beta 40 in APOE3 mice and APOE4 mice. On panel B are shown results of ELISA for A-beta 42, and again A-beta 42 was decreased in APOE3 mice and APOE4 mice. The conclusion is that bexarotene treatment decreased soluble A-beta level in the interstitial fluid of APP mice expressing APOE3 and APOE4. Lastly, we measured the effect of bexarotene on HDL level in plasma of the same mice. And here is on A is shown the results for APOE3 mice. You can see that bexarotene increased the level of HDL in the plasma of APOE3 mice approximately by 13% compared to vehicle-treated mice. Similar result was received in APOE4 mice. HDL level was increased by approximately 50% by the treatment compared to vehicle-treated mice. So the conclusion from this study is that RXR agonist bexarotene improves memory deficits in APP mice expressing APOE3 and E4 without changing amyloid load. Bexarotene decreased soluble A-beta in ISF, in interstitial fluid, suggesting that it improves clearance. And finally, bexarotene increased plasma HDL in both genotypes. On this slide, I'm showing what is our hypothesis for the roles of ABCA1 and APOE on A-beta metabolism. As I mentioned at the beginning, A-beta is secreted as monomer by the neurons. It can form oligomers, and they eventually could convert to A-beta fibrils, and they can form plaques. The role of ABCA1 and APOE is to regulate cholesterol efflux in glial cells, where lipid-poor APOE obtain cholesterol and phospholipids and convert in lipid-rich APOE. As I mentioned in the beginning, one consequence is that lipid-rich APOE is a longer lived than lipid-poor. Another consequence might be better binding of lipid-rich APOE to A-beta, which eventually can affect A-beta aggregation, decreasing the aggregation and preventing the formation of oligomers and fibrils. The consequence of this is that soluble A-beta, either as monomer or smaller oligomers, can have better chance to pass blood-brain barrier. Another consequence is that soluble A-beta could be better degraded or easily degraded by extracellular proteases or phagocytosis by microglia. And eventually, all this can decrease the level of A-beta in the brain. Finally, in plasma, increased level of HDL could increase the binding of A-beta to HDL, eventually leading A-beta to be cleared from the body, which decreases plasma concentration of A-beta and affects the balance between plasma and brain and stimulate peripheral sync of A-beta clearance. So finally, I would like to thank my collaborators, Dr. Lefterov, who has been my collaborator for the last 25 years, Dr. Nicholas Fitz, who performed most of the studies. Andy did FPL studies and Western blots. Emily and Alexis performed the behavior of these mice, and Pratyusha and Moose did most of the A-beta and ELISA data. These studies were supported by grants from NIH, Defense Department, Alzheimer's Association, and ISOA. Thank you for your attention. Okay, so there is one question. Okay, the question is, when were the mice treated with bexarotene? Was it chronic? The mice were treated at 7 months of age, which is when they have fully developed plaques and behavior deficiencies, meaning that the treatment was therapeutic. It was acute treatment, only 2 weeks. Bexarotene was given only for 2 weeks by oral gavage. I don't see any other questions. Okay. Where is this slide? Okay. 1 second.